everyone, welcome to Vedantu NCERT Solutions 11th and 12th. I am Anshana, your biology master teacher, and today we are going to discuss question from the chapter called photosynthesis in higher plants. First question, by looking at a plant externally, can you tell whether a plant is C3 or C4? Why and how? Okay, so we need to tell by looking at the external feature, that is morphology. By looking at the morphology means leaf, stem, root. By looking at those features, we need to tell whether it is a C3 or C4. Can we do that? The answer is no. We can't say a plant is C3 or C4 just by looking at the external feature or morphology. We need to see the internal feature, that is Kranz anatomy is present or not, or dimorphic chloroplast is present or not. We need to check that. Okay, clear, right? By looking at which internal structure of a plant can you tell whether a plant is C3 or C4? Explain. Okay, so here they said clearly that by looking at which internal structure, the internal structure which is present only in C4 plant is, okay, first one is Kranz anatomy. Kranz anatomy is present only in C4 plants. And one more thing is dimorphic chloroplast. Okay, dimorphic chloroplast. So these two are the very important key characteristic of C4 plant, which is present only in C4 plants. Okay, so in C3 plants these two structures are not present. So hence C4 plant can produce more amount of photosynthetic products than the C3 plants, because C3 plants have lot of wastage such as photorespiration. Clear, right? Okay. Even though a very few cells in C4 plants carry out the biosynthetic Calvin pathway, yet they are highly productive. Can you discuss why? Okay, so in C4 plants there are mainly two characteristic feature. Okay, two main thing happening there. What is that? One is Kranz anatomy. Kranz anatomy means what? Kranz anatomy means surrounding the vascular bundle there will be a bundle sheath cells. Okay, so this bundle sheath cells having chloroplast which is do not containing grana. Okay, so these chloroplasts do not containing grana. So hence the second key characters of C4 plant is dimorphic chloroplast. Okay, dimorphic chloroplast. See here the bundle sheath cell do not containing grana, but the remaining mesophyll tissues containing chloroplast which is having granas. Okay, so hence there are two type of chloroplast present in the C4 plants. So these two structure helps the Rubisco to produce that is glucose than carbon dioxide. So the Rubisco is the enzyme which can actually act as carboxylase enzyme and oxygenase enzyme. In the presence of high temperature and concentration of oxygen, more concentration of oxygen, resulting this enzyme to act as a oxygenase enzyme. Hence photorespiration is going to happen. But these two key characters, these two structure helps this Rubisco to reduce the temperature, means that surrounding will be reduced temperature, and also there is no oxygen in the bundle sheath cells. These are the bundle sheath, right? In this place there is no oxygen, hence this area containing Rubisco enzyme will produce only glucose than carbon dioxide. Clear, right? Rubisco is an enzyme that acts both as carboxylase and oxygenase. Why do you think Rubisco carries out more carboxylation in C4 plant? Okay, so it is a very direct question. You can easily answer this. You know, Rubisco means Rubisco means it's an enzyme which act as both carboxylase and oxygenase, right? Rubisco means ribulose bisphosphate, carboxylase and oxygenase enzyme. That means this enzyme can act as carboxylase enzyme and also oxygenase enzyme. In the presence of more concentration of carbon dioxide, less temperature, this act as carboxylase enzyme. This enzyme can act as carboxylase enzyme. But in the absence of carbon dioxide or more concentration of oxygen and high temperature, this enzyme is act as oxygenase. Okay, but in C4 plants, it act as this Rubisco helps in carboxylase alone. Why? Because we need to understand the anatomy of C4 plant. See, this is the outermost epidermis. Here, upper epidermis. Below it, we can find mesosomes, and here is the vascular bundle, and here we can find bundle sheath sac. Okay, so here in this area, this mesophyll tissue is having more temperature because it is directly exposed to sunlight. So here we can find more temperature, but when it is going inside in this area, the temperature is less, right? Okay, one more thing, in mesophyll tissue there is no Rubisco. This area there is no Rubisco enzyme. The Rubisco enzyme present in the chloroplast of bundle sheath here. Okay, so hence the carbon dioxide is fixed here, that will transported to bundle sheath. In the bundle sheath, what happens? The dark reaction is going to happen. So there is no oxygen, less temperature, so that we can find more amount of glucose, that is dark reaction product. Clear? Yes. Suppose there were plants that had a high concentration of chlorophyll B but lacked chlorophyll A. Would it carry out photosynthesis? Then why do plants have chlorophyll B and other accessory pigment? Okay, so if there is no chlorophyll A, photosynthesis happens. Check with it. Can we find photosynthesis without chlorophyll A? No, we can't. Okay, so chlorophyll A is the reaction center. From here, the electron have to transport it to high means it has to exert it, right? So if there is no chlorophyll A, then there is no first reaction, that is light reaction. When there is no light reaction, there is no dark reaction also, right? There is no photosynthesis at all. Okay, then why this chlorophyll B and chlorophyll C or phycobilin and carotenoid, these are all called accessory pigments. Those pigments are called accessory pigments. What is the function of those things? Even though if it is not present, we can find photosynthesis, but this molecule chlorophyll B, okay, and xanthophyll, carotene, these are called accessory pigment that also capture the photon and convert that energy or transport that energy to the chlorophyll A. So it helps to trap the energy from the sunlight. Okay, clear, right? So it is not going to produce photosynthesis or light reaction. These molecules that is accessory pigments, that is chlorophyll B and so on, right? So chlorophyll B, chlorophyll C, D, and even carotenoids, xanthophyll, these are all helps in the trapping the energy from the sunlight and transport that energy to the chlorophyll A. Okay, why is the color of a leaf kept in dark, frequently yellow or pale green, Which pigment do you think is more stable? Okay, so when we take a potted plant and kept in a dark place, after a few days we can see yellowish color in the leaf, right? Yellowish color in the leaf. What happens? Why it is yellow? Why it is yellow means the carotenoid or carotenoid pigments are more stable. Okay, carotenoid is helps in giving the plants to which color? Yellow or orange color, right? So this yellow or orange color is given by carotenoid. That's why it is called more stable. Clear? Look at leaves of some plants on the shady side and compare it with leaves on the sunny side, or compare the potted plant kept in a sunlight with those in the shade. Which of them has leaves that are darker green? Why? So we are keeping two pots, one in the direct sunlight, another one is in indirect sunlight, that means under shade. So which one have more darker green color? Which one? Have you checked this one? Have you looked it or experimented this one? No. Then we can say we can experiment. You can experiment actually. That is the shady that the plant which is kept under shade have more darker. Why? Because in the direct sunlight the temperature also more, so due to temperature this plant is not going to perform photosynthesis properly. Okay, but this plant under shady places can do more photosynthesis because it is getting light, but it is in the shade places, so temperature is also less. Okay, question number 8. Figure 11.10 shows the effect of life on the rate of photosynthesis. Based on the graph, answer the following question. Question number 1. First, A. At which points A, B or C in the curve is light a limiting factor? Okay, so here is the graph, right? So here there are 3 points, A, B and C. In which point, okay, in which point or which curve light is a limiting factor? Where we can find light is a limiting factor? If light is stable, then we can find more photosynthesis, more amount of glucose, right? If the light is limited or the light is very less, what happens? The photosynthesis is also less. Hence, we can easily tell that A and 50%, right? 50% of B is a point where light is a limiting factor for photosynthesis. Okay, second one. What could be the limiting factors in region A? So light is one thing, right? So light is one thing. Along with the light, carbon dioxide. Okay, so if the carbon dioxide is also less, then photosynthesis is also less. And water. If water is less, then photosynthesis is also less. If temperature is more, photosynthesis is less. Okay, so these are the limiting factors in the region of A. Okay, next one. What do C and D represent on the curve? Okay, the point C represent, here the light is stable, right? So light is also stable and photosynthetic rate is also stable. Okay, it is going in constant way. Even though the light is more or less, okay, if the light is more or less, in this point the photosynthesis is stable. And D, the point D represent beyond this limit. Okay, beyond this limit, if the intensity of light is more, then there is no change in the photosynthesis. Clear, right? Give comparison between the following: C3 and C4 pathway, cyclic and non-cyclic photophosphorylation, anatomy of leaf C3 and C4 plants. Okay, so these 3 points we need to distinguish. Okay, in which first one, let's discuss first one, that is C3 and C4 pathway. In C3 pathway, rubulose bisphosphate is primary acceptor of carbon dioxide, right? So the Rubisco is present, right? So this Rubisco is the enzyme which combine carbon dioxide and rubulose bisphosphate. Okay, so primary acceptor is rubulose bisphosphate, and it is going to produce phosphoglyceric acid. Okay, PGA, 3-PGA. And in C4 pathway, the primary acceptor is phosphoenol pyruvate. Okay, phosphoenol pyruvate, and the product which is produced in this C4 pathway is oxaloacetic acid. Okay, oxaloacetic acid. Next point is the same thing that we are already discussed, that is 3-PGA is the first stable product in these C3 plants, and in C4 plant, oxaloacetic acid is the first stable product. Okay, next one. It occurs in the mesophyll leaves, mesophyll cells of the leaves. Okay, so this reaction, the first reaction called rubulose bisphosphate is combined with carbon dioxide and producing the PGA happens in the mesophyll tissue. This is also the first reaction, that is PEP is going to combine with carbon dioxide and producing oxaloacetic acid happens in the mesophyll tissue, but the Rubisco is present in the bundle sheath. So this reaction happens both in mesophyll and also in the bundle sheath cells. Okay, next one. The process of carbon fixation is slower in C3 and faster in C4 plants. Okay, next one. Cyclic and non-cyclic photophosphorylation. So we need to distinguish cyclic and non-cyclic. In cyclic photophosphorylation, only photosystem 1, that is P700, helps in photophosphorylation, but in non-cyclic photophosphorylation, photosystem 1 and photosystem 2, that is P700 and also P680, both helps in light reaction. Okay, so next, here the product is ATP alone. In cyclic photophosphorylation, ATP alone is produced, but in non-cyclic photophosphorylation, along with the ATP, NADPH2 also produced. Okay, so next, here in cyclic photophosphorylation, only photolysis, that means splitting of water. Okay, photolysis means splitting of water. There is no splitting of water, but in non-cyclic photophosphorylation, we can find photolysis, that means splitting of water into 2 proton, 2 electron and nascent oxygen. Okay, nascent oxygen. Next, electron move in closed circle, that means from the chlorophyll A, electron is excited to the higher energy level, and this electron is transported back to the chlorophyll A itself in the cyclic photophosphorylation. But here in non-cyclic photophosphorylation, the chlorophyll A is release 2 electron that will move to high energy level, and from there it will transport it to electron transport system, and finally this electron helps in the production of NADPH2. Okay, so there is no returning, so hence it is called non-cyclic photophosphorylation. Yes, okay, so next one, anatomy of leaf C3 and C4 plants. So we need to distinguish between C3 and C4. See, in C3 plants do not possess crans anatomy. Crans means what? Wreath, right? So the wreath-like structure which is present around the vascular bundle. So this is the vascular bundle, means around the vascular bundle we can find bundle sheath cells, which is present only in C4 pathway. There is no bundle sheath cell present in C3. Hence, we can say no Cran's anatomy in C3. Cran's anatomy is present in C4. Okay. Next, chloroplast is not dimorphic. That means only one type of chloroplast is present, that is grana. The chloroplast which containing grana. Okay. But in C4 pathway, we can find both grana and the chloroplast do not containing grana. Okay, so agranal chloroplast and granule chloroplast, both we can find. Okay, next one. Mesophyll cell possess intercellular space in case of C3 plants. Okay, so this is the outermost epidermis. Below the epidermis, we can find palisade parenchyma and spongy parenchyma. In between, we can find more amount of intercellular space, but in this particular plant, that is C4 plants, there is no intercellular space. Okay, so this is about the chapter photosynthesis in higher plants. Hope you have understood each and every question. If there is any doubt, please comment below, and if you like the content, please like our video and share it to your friends and subscribe. Thank you all. Bye.